All right. So, welcome to the first of our feature series at CCL. And today we're going to be talking about the BioArtBot and Distributed Cloud Lab, um, sort of very briefly, because we talked about it a little before already. This is a project that's been going on for about a year and a half. My name is Tim Dobbs, and I've been the sort of constant touch point for the project. But has been a group of various people who have contributed all kinds of skills. Um, Including the idea for this cool concept art, which is really neat. So this has been kind of a collaborative effort through a lot of different people, and we're going to present kind of where we are right now. So, how can humans talk to microbes? This is our kind of TED Talk, kind of like a thought leader kind of question. But it has some legitimacy to it because we have all these microbes all around our world, and it would be nice to be able to interact with them. Um, so if we want to kind of ground that question a little more strongly, we can say, well, how do you make it possible for anybody to express themselves using biology? And luckily, there's already a tool set that's been thought about and developed for more than 50 years, which is websites. Um, Computers in general have done, or computing, the field of computing in general has done a lot of work on figuring out how do you make things that are easy for humans to interact with. How do you make user-friendly design elements? And once we've kind of got it into this website form, an idea of any kind, we can then send it to a robot because now it's all sort of computer code. And there is one thing that robots are really good at, which is that they kind of are good at interacting with the physical world. And specifically in this case, it would be biology. So we have a robot here that can move liquid around that has microbes in it. And we can program the microbes themselves to, in this case, produce a bunch of different colors. And then that allows us to, if we follow the whole chain through, we can see that you can have an idea for a drawing and then tell the website about it. And then the website tells the robot, and the robot instructs the microbes, essentially. So we've kind of created this bridge to the microbial world. Um, and if we want to take this in specific, so that's kind of the broad generalities. Here's how we did it in BioArtBot. So I'm just going to kind of jump to the end of like how the whole process has worked. We built this platform. It's at bioartbot.org. And it's just a place where you can make drawings. And then we got these E. coli, and they produce colors. And then both of those feed into this robot. And then in the end, you incubate it for 48 hours, and you get this really cool E. coli drawing of Pikachu. um, so that's pretty much the process. There's a lot of honing and zeroing in, but this is kind of how it works. And so this way, anyone from around the world can go to this website, make a drawing, and hit submit. And then within a couple of days, depending on when we are able to run the robot, they get back a photo of this very cool thing where their art is actually growing. Sometimes we call it microbial ink because it's colored microbes. Um, and so it's really neat. And we had a really good experience with our first pilot run in 2019, which you can see here. We had about 35 drawings submitted from around the world, and we printed them. It was really an exciting time for me. It was a lot of work getting the kinks worked out, but we got at least 4 countries that I can think of. We didn't really do a survey of the users, but just from like emails I recognized or people I knew through, contacts of contacts, I was like, oh wow, this is actually kind of global, which is really fun and interesting. And also something that I found really exciting was a biology class reached out to me, and at least a few students were able to submit their drawings for part of their like AP Bio high school class, which was really cool. So that was great. This was in fall 2019, and it was kind of early 2020 by the time we actually had all the prints done. And then it took a little longer for pandemic reasons to actually get it all back out to everyone. But this was a really good, successful pilot. Um, so then that's cool and great. But then if we emphasize, well, how do you make it possible for anybody to express themselves with biology? We can start to think about like, okay, is this just like a fun, neat project, which it is, or what does this does this project have any sort of greater calling? And if you start to look into the literature, you can see that there's two kind of paths that I have seen kind of come up fairly regularly in biology education or synthetic biology education. One is this concept of kits. So this is the BioBits Bright. It's just one of a number of kits that are kind of circulating out there, which is the idea is they're these sort of low cost little packages that can be sent out to schools or anyone who wants to learn biology, and they're able to sort of do a science experiment hands-on in a sort of semi-controlled way. So they still get to have the fun of the experiment, but they don't have to, have a whole biology lab or be, someone who's really good at designing experiments, for instance. This is a great way in. The second one is, there he goes. This idea of using robotics to actually help. A lot of work coming out of this lab at Stanford here. Well, basically, the idea is we have sort of telepresence in a lot of other aspects of life. We've all been on Zoom, I think, a fair bit in the past year. Uh, but the way robotics works is it lets us run experiments in situations where we might not be able to. So maybe I don't have access to a BSL-1 lab, but I do have um, a computer and a web connection. And so I can talk to someone who does have that access, and then you can do some neat experiments in a using like sort of controlling a robot remotely. So we see that this works pretty well. So how can we translate this to what we've been doing? Well, hopefully this connection starts to make sense. I've been calling it this kind of walled garden learning model. And it's this idea that BioArtBot is this like fun little controlled space where, yeah, you make a drawing and it's nice. And we kind of make that first quote unquote experiment really straightforward. It's think of a drawing you want to draw and then draw it and see if it can grow in this really standardized way. Um, so that's nice, but it's a pretty limited process overall. The kinds of pictures you can draw they tend to be pixel art. If you look at everything that we've ever done, a lot of people choose to do video game characters, which I think is really fun. But I think it's because of the pixel art kind of look that draws them that way. Um, so what if you wanted to use kind of broad strokes, or what if you wanted to try to recreate a Van Gogh painting in microbes? That would be pretty cool. Um, there are ways that you can sort of take BioArtBot and expand it from there. So you can say, um, uh, okay, yeah, I want to do big broad strokes. So I have to think about the robotics. How do the robotics work and how do we actually apply microbes onto the ground? Or maybe, all right, well, I just want to upload Starry Night to a website and I want it to be grown on microbes. Sure. Okay, well, that's a software engineering problem. How do we think about that? Or maybe you say, man, there's no purple here. Or maybe you say, man, the teal color that CCL has really stinks. Because my nemesis is the teal colored strain because it just never produces the protein enough. Um, and so maybe you want to think about like, okay, well, how could I get a better teal? Or how could I get purple instead? Or something like that. Great. Now we're thinking about microbiology. And so there's this model is basically, oh, it's a very nice, pleasant garden to be in. It's a great place to start. But there's all these sort of paths that you can walk down and sort of be subtly suggested that you might be interested in that go to sort of these bigger, scarier forests that I've represented here with silhouettes of trees. So this is the idea. And I think we've kind of shown that like, yeah, this is the starting part works. But if we want to kind of expand it and we want to really make it something that like, okay, but how can anyone do this? It's not just something running out of CCL. We've built it as an open source platform for that purpose. So you can see here, I've got this little, as seen on TV, star here in the bottom that we're all open source. And the reason for that is that it's low cost and it's accessible. We want to make it so that anybody can get into this. And so it's all built, the web stuff is all Python and Flask, which is pretty accessible and all free, of course. And then the hardware we're using is the Opentrons. We're not married to the Opentrons, but it is a good, solid, mostly open source system. So that's why we like that. And then as far as strains, we want to try to use everything in OpenMTA. There's a great culture of sharing freely strains that are under OpenMTA, and so the idea is to try to keep those things, um, you know, accessible. This should be something that anyone can pick up for not that much money. Um, so there was a second reason to do open source. Again, kind of coming along that idea, how do you make someone access that walled garden really quickly and easily, and then be able to do kind of those bigger adventures. And the second idea is this flexible engagement model or progressive engagement model, which I've sometimes called it. Uh, and the idea is it's sort of this arrow of you can start at the beginning where you're just sort of initially exposed. You just submit a drawing, and then we'd like to include some sort of discussion modules. We don't have anything like that in the project right now, but I think it would be excellent to sort of pair it with like, okay, so why did the things make the color? Let's talk about the biology at work here. Let's talk about what the robot is actually doing. And so that way someone gets back a nice photo of a drawing, and that's really cool. But then they can also kind of spend another hour, maybe in a classroom context, or not if they just want to read about it, sort of saying, okay, why did all that happen? And so that's great. I mean, that's a great start. It's a hands-on experiment for people interested in kind of getting started. But you can also go a little deeper. So perhaps we have some modules that suggest a way to change some code or biology. Okay, do you want that purple? Okay, well, here's a plasmid you could order, and here's a method for doing it, and now you can have purple E. coli as well. If people are really interested, we're moving past. I would say the deeper look is maybe sort of, kind of at that more advanced level of a classroom environment. But taking ownership is kind of a part now, where you're kind of moving into more of a maker situation, or a place where you say, I really want to get. I'm loving this, and I really want to learn something, or I want to start making something. So you could say, I've got an idea too, and I'm doing this on my own robot, so why not just change the code myself? It's all open source. I'm just going to see if I can make something happen. And then finally, at the end, is this kind of really broad vision of joining a global community. So the idea is that, like, oh, well, this is all open source, and if multiple people want to have their own versions of this going, we can all sort of get together and have a process for sharing code and sharing biological information. And so that's a way in which this allows someone to go from being a student who just thought it was kind of fun to make a drawing, all the way to someone who's sort of part of the larger DIY bio community. And so that's really exciting, and that's really why we've made a real effort to keep it, as open source and as cheap as possible. I've gone ahead and made the same arrow in meme format, uh, where the sort of smooth brain format here is you make a submit to you submit to this BioArtBot cloud, you get your drawing back. Hey, that's really great. And then we go all the way up to this idea of Galaxy Brain being a part of an open source distributed bio cloud lab. And so we'll touch more on that a little later, but the idea is basically like, yeah, we can all sort of take these concepts and use it as a shared collaboration venue for doing DIY bio. So that's exciting. Okay, cool. All right, so what did we actually do? That was a lot of like metaphors and a lot of diagrams and stuff. And I think before I go any farther, the next thing I'm going to jump into is okay. So how did we do it, and what exactly are we doing with all this stuff? Just to kind of give you a little taste of that. But before I do that, does anyone have any questions? Any thoughts before we go any further?
Yeah, did the pilot program, did everyone just submit sketches, or were they using Protocol Designer or anything else? Was it just sketches?
It was all done with something that looks a lot like the bioartbot.org website right now. So it's basically a grid where you click in parts of the grid and it makes it colors. Cool. All right. So the first part is software, if we're kind of thinking about this. So I've been having a lot of fun. I realized I can make GIFs in Google Slides. So I've been doing that a lot. So here it's like I said, it's a grid. You click, you pick your color, you type in a name and an email, and then there's a small workflow for like making sure you're a real person. So that's kind of part one of it. And then this is the second part, and this is a really, really early prototype. We've actually been doing most of this on the command line. Um, and so this is early prototype for what a graphical user interface for getting the art looks like. So here I've got a grid of everyone people have submitted. I check all these checkboxes to say, yes, I would like to print these on the robot now. And then you hit download print protocol. And what happens is it translates that into robot code via template. And then we just pop that on the Opentrons, and the Opentrons is able to print these. So so those are our kind of big user interface pieces of software. So what this ends up looking like, again, in sort of diagram mode, is we have this drawing UI where you draw, and we have a printing UI here. And then on the background, there's this web server that runs, and it has to be really good at kind of organizing the data and making it all make sense. And then finally, it all gets pushed out to this robot code, which has to be good at talking to robots. And so then the robot kind of slides in here, and we send that information onto that robot. I wrote here at the top, keep it simple and flexible. So that's really our big challenge here. It's not a totally crazy piece of software to write, right? It's not, we're not mining Bitcoin or, I don't know, doing any particular crazy machine learning. But the goal here is to have both of these things, simple and flexible. And sometimes that's really hard. It needs to be simple so that it's the sort of thing someone can jump in and learn on, but flexible because it kind of has to do a couple of different things, as you can see here. And we don't want to limit it to being only ever able to do printing pixel robot art, right? Maybe we want to do different kinds of art, or as we'll talk about a little more in the future, any arbitrary lab protocol. So that's the challenge there. I've actually been having a lot of fun. I'm not a software engineer by trade. I do a lot of software stuff, but I've been learning a lot, and it's been a real kick for me learning how to make like a really robust web app that can kind of handle a lot of the complex situations that pop up on the internet. So the next thing is the strains. So we've got 5 E. coli, and they have plasmids in them that do that you might call reporter plasmids to say, yes, I'm expressing the right gene. In our case, we really only care about the color that comes out. Often in microbiology, you would say, I see that it's glowing green, which means that my insulin gene has turned on. In this case, we're really only interested in the colors. So we have 5 of them, and I've put there 1 bad because my nemesis teal is still in our set until we can figure out a better one. And I'm really interested in something that actually Patrick suggested originally, but is some really great work by the people at the Free Genes Project and the Open Bioeconomy Lab, which is a set of 42 different reporter genes, one of which is here. This is a blue that is available for free through the Free Genes Project, and it's called the Open Reporters Project. So I would really like to get that, get that in and start putting that into strains so that we can kind of, have a much broader palette. I don't know how many of those 42 would actually look interesting, but, probably more than a few. 42 is quite a few, but it'd be really cool to have like a 96 well plate just full of different colors so you can really do interesting stuff. And we've also talked a lot about trying to look at just different ways of handling expression here. There hasn't been a ton of work on this, to be honest. But it's definitely something that we're thinking about in the next phase. It's like, okay, well, what about how could we change the copy number in a plasmid to produce different intensities of color if you wanted to do some shading or something like that? Or it could also be done, how do you make the biology interact with itself? Maybe it does some kind of quorum sensing, so it can kind of be one of the artists in the piece of work. And then this is just a short video showing like, okay, well, this is the actual like work that we've been doing. And it's a lot of, growing cultures, growing mediums and moving around, which is actually really perfect for getting hands-on in biology. I think kind of the secret to all this is that a lot of us in the project are sort of learning at least one of these skills as we go. So this has actually been really great for figuring out how to do basic stuff in a biolab, like grow cultures and then passage cultures, keep them alive, and then do sterile technique and stuff like we're seeing here. So yeah, here we're just kind of loading each color. You can kind of see they're labeled there into a plate, which will eventually go on the robot. And so it's like preparing a palette. So this is great. We're kind of working with the strains, and I think we'd like to really engage more with the kind of strain aspect of it as the project moves forward. So I'll just jump ahead. I don't think we need to watch every single step. So then there's the robot. So we talked software, we talked biology, and then there's the robot. This is our Opentrons. It's an Opentrons 2, and we've been using we've been going back and forth. The 10 microliter pipette is just a little more precise. But basically, I'll hit play here, and you can kind of see how it works. We have all our colors up here. These are cultures, liquid cultures that are growing. And then the robot goes, and it picks up some of that liquid culture, and it just touches every single point in these square agar plates. And what happens there is it basically just inoculates a colony in that spot. And then after 2 days, a colony will grow. So it's really, really fun to watch because you don't see anything at first. You're like, sometimes I'm like, well, I wonder what this picture is. I know the name of it. It says, Jack. All right, I wonder what that is. And then you grow it and it turns out it's a pumpkin. So it's really neat to see that happen. I'd really like to share that experience with the people who submit. And so I've been thinking for a while now, we've been talking about getting some kind of time-lapse growth, video in place, because I just think that would rock so much. And so what are we playing it again? Come on, Google. Okay, great. So this is another video where as far as like how do you work with the robot, it's we had to program the original protocol, which then gets slightly changed for every different piece of art that you print. But there's also a lot of really kind of fun efficiency stuff. So this is a before and after. Originally I had a pretty inefficient protocol written, and you can kind of watch it print here. It's sort of like puts it in, waits for a little while, moves, does another one, and then I figured out how to make it a lot more efficient. By just sort of changing the kinds of instructions. And so you can see here, it moves much, much faster, and it's a little closer to like a sewing machine, which I really like when you can just kind of watch it go. So that's really neat. And I think there's actually a lot of room for further optimizations here. We've kind of written this one master recipe for a robot, but it can definitely go a lot further. And especially if you start to think about like, well, how do people draw? So often people like to have a big blue sky. Okay, well, if we often have 150 blue dots next to each other, is there a way to do that more efficiently? So these are the kinds of questions we've been working on for the robot itself. So that's been a lot of fun. And like I said, we're learning as we go. I kind of drew this drawing originally almost because it was my experience. I don't. My formal training is actually in chemical engineering, which is none of these things you might notice. And so it's been really fun, kind of, peeking my way into each of them as I go. And that's been true of all of our contributors. We have some people who come in, they're software engineers who are just like, yeah, no, I know about JavaScript, and I'm interested in biology, and this is kind of a way in. Automation engineers, same thing. Maybe you want to learn web development or whatever, and then the microbiologists as well. And so far, it's been a real way for people to kind of branch out, which is really exciting. And here's an example of our learning process. This was our very first test print. It was supposed to be a smiley face. It looks sort of demonic. And then you can see this Pikachu. I didn't draw this. I actually don't remember who submitted this originally. It was just, someone from the internet. And it's one of my favorite ones. I just think it's so neat, and it looks like a Game Boy. And I want a biology Game Boy now. And so, yeah, this is the process, and it's been a ton of fun. Okay, so I think I really just have one more section, but this might be a good place to stop and check in on kind of all that more nitty gritty stuff. Does anyone have any thoughts or questions on that? I'm seeing Octoprint. I don't know anything about that. I'll have to look into that.
For the colors, are they all visual colors or some of them UV? What's the reporter systems? Are they just color?
Yeah, they're Mostly chromophore proteins. So they're proteins that just sort of have, a visual, you can see it with the human eye. GFP is one of them, so green fluorescent protein. And that one you can see without black light, but it looks way cooler with the black light because it kind of glows. And you can see that in some of our photos that we've taken. We have links to all of the sort of BioBricks pages for every protein that we use, which I think is really cool, and I want to kind of keep doing. And what I'd like to do eventually is get on the website a place where you just hover over the color and it says, you're drawing with this protein or this plasmid. But right now I don't really remember all of them. There's a blue that's really good, and that's the one I always use as an example because it comes out so so visually striking. But I have the link to that somewhere. But it's a specific kind of blue, I think, from a jellyfish, or maybe it's a coral. I don't know, something in the ocean. But yeah, mostly
the five E. coli strains that we used were the ones that were distributed as part of the first time that ASM did the agar art competition with the community labs, and I believe they may have been generated by somebody at Bugs. So they are just E. coli with some of the chromoprotein BioBricks. And I believe actually all of those are listed or also on the Open Enzymes reporter spreadsheet that I had posted earlier. So let me put that in the chat here again as well. So there's that list contains 8 chromoproteins that are off patent, 30 fluorescent reporters that are off patent, and a couple of other things like bioluminescent genes and stuff like that.
Cool, thanks.
Cool.
Corinne had put a couple of questions in the chat too.
Yeah, I see that. Corinne, you say, can you remove agar from one petri dish and place on another print and have images grow together into 3D layers? I don't know, and that's a very exciting idea. Yeah, we've been bouncing around ideas about how to make things 3D. I think there's a lot of opportunity there because it's pretty simple. Yeah, and so far we really haven't played with it too much. It's been kind of just like, well, how do we get it kind of to this first set of standardized platform? But I love that idea, and I think it's definitely worth exploring. I think at some point it's probably a good idea to have a spreadsheet of like, here's all the crazy ideas that we've ever come up with.
Yeah, Corinne, I would love to explore sort of alternative ideas working with agar. We may want to move away from actual agar because agar gels tend to be kind of brittle. They tend to break if you manipulate them. But there's other hydrogels that are a lot more flexible, and you should be able to unpeel from a petri dish and just stack on top of another petri dish.
You could also imagine actually like setting the robot to pierce the agar and have it grow within it, and then another layer on top too.
Yeah.
Yeah. Well, so strictly speaking, it does pierce the agar very slightly, and that's just because it's hard to guess where the actual surface of the agar is. And so we just overcompensate and we do pierce it a little bit. But we haven't played with like using that to our advantage. It's just kind of, this is the best we could do. But I think that would be very cool. And maybe we could make new shapes. I mean, that would be really fun, right? Does it need to be a totally flat plane? Maybe not. So yeah, moving on in the questions. Excuse me, Tan asks, how do you originally collaborate with the bulk of this project? In person, Mattermost, or Slack, or email? So originally it was through meetups at CCL. Obviously, we're not going into CCL at this point. But I'm looking to figure out a good way to sort of continue that, maybe do a regular meetup again. I kind of stopped mid-pandemic just because, I don't know, it was hard to make forward progress, but I think we're kind of in a place now to kind of keep going on that. The other way that we sort of collaborate now, there's some email, but a lot of it runs through GitHub. So all of this code is in a GitHub repository, which I'll flash up at the end, and we can also find anywhere. And we use GitHub issues pretty extensively, so it's kind of a way of doing organization. It's obviously GitHub is built for code, but it doesn't necessarily need to only be code. So some of our issues are things more like playing around with strains or something like that. And the idea is you sort of do that communication through working on an issue, and then you push to that repository so everyone can see your work when you're done.
Tim, would you mind explaining what GitHub is for people who might not know?
Yeah, of course. Thanks for pointing that out. So GitHub is a tool that's used primarily for collaborating with software, but essentially what it is it's a place to keep all of your documents on the internet, so at github.com. And there's like a corner of github.com that is like the repository for all the documents around BioArtBot. And what it does, and the reason it's better than just like a shared Dropbox or something, is that it tracks every change, and it has all kinds of tools for helping people collaborate. So it says if, for instance, if Rami says, well, I'm looking at this code and here, I want to make this small change. I think it might make things better. Then she can submit it to that, and it sort of automatically emails anyone who's flagged, so maybe me, and they says, hey, can you review this and double check that it all makes sense? And then I look at it and I go, oh yeah, that makes a lot of sense. And then with one button, I can merge it into the existing stuff without worrying about any changes.
I think another aspect of it, Tim, having it on a GitHub repo or a repository, so where everything is, all the information is gathered, is, I mean, I know GitHub itself isn't open source, but if you have all that information there, and we're talking about like decentralized models that other people can replicate, you can easily do what's forking the code. So you just take this thing that everyone has collaborated on, and let's say I want to make a different version. I can use the skeleton and the model that the that the people came up with, and I'm just like, basically it's like a generative thing, Corinne, if I may point this out. It's sort of like, yeah, it's literally a forking of the code, but it's almost like, I think of it as like sort of like a cut and paste kind of thing. And then when you're modifying code, and you're just like, which can get messy too, but I'll just like leave it at that. But that is sort of like the point of, I think, Tim, the distributive model that you're also looking for, which allows that to happen. That documentation is so important when you're looking at like open source and you're wanting to collaborate with other people.
Yeah, exactly. Thank you for highlighting that. That's actually really important. The idea is that, we put this out in the world, we're building this idea, but really anyone can take it and do anything they want with it. My dream is that lots of people take it, and there's kind of lots of different flavors of it kind of working around out there. And ultimately it's, sort of supporting people in their kind of DIY science needs without without things getting deeply confused, because it's still possible to kind of see the documentation and kind of see where everything came from originally. So there's a pretty good paper trail that Git sort of makes automatically. Oh, and just for the sake of, not confusing anybody, Git and GitHub, GitHub is just like a specific version of a thing that does Git is the sort of generic version. But if I said one or the other, I just don't want to confuse anyone. They're effectively the same thing in this context. Great. Yeah, I'm seeing more ideas about materials, and I think, yeah, let's definitely keep talking about that. But I think at this point, I'll kind of move on. I've just got a few more slides here, so we can wrap that up and then see where we go. So you might remember this question: how can humans talk to microbes? A sometimes more compelling question is: how do humans talk to other humans? Collaboration is hard, right? Especially kind of as you start to think about these kind of big, expansive ideas, like Git is supposed to help it so that, someone in, I don't know, Missouri can take a look at what we've been doing here and say, oh, okay, I basically get it. But that's not trivial. So how do we help support that kind of thing? I'll come back to my meme here. The vision, as it kind of comes from this thing that we've been calling BioArtBot, and moving it into like, oh, what about this distributed cloud lab concept? Is that, well, you could take this idea and it could be your venue for collaboration. So the way Git is really good at helping you collaborate with documents or sort of just like project work, you could start to imagine that BioArtBot or its little brother, Distributed Cloud Lab, could be the sort of thing that could help you collaborate around scientific protocols. So if you kind of visualize, and at this point, I'd been having a lot of fun all day making slides that had like fun animations and stuff. So at this point, it actually gets pretty simple because I was like, oh boy, I gotta get dinner and get to this meeting. So you can kind of imagine this is sort of the workflow right now for BioArtBot. So someone here has this idea for a drawing, and then they send what is a pretty standardized drawing protocol. So the reason the whole thing works is that all you have to do is use this design software, this little design app with the grid that you click, and it's translated into something that's quite standard. So it's easy to replicate. That goes up to our BioArtBot design website, bioartbot.org, and then it just gets pulled down and sent to a robot. So great, that's really great. You can imagine sort of making that more abstract and generic, and instead saying, we have a distributed cloud lab design website and a standard just BioLab protocol. So something that's pretty well defined. You probably couldn't do like a really, really out there experiment without, getting a lot of people on the phone. That has been my experience. Although I'm excited about the idea of being able to make this stuff a lot easier. But if I'm somewhere that doesn't have access to a lab, or maybe I don't have access to a robot, but I'm doing some work in my lab, or maybe I just want to do a really big experiment that needs more than one robot, and I can kind of run them in parallel. You could start to think about designing a protocol that goes up to some kind of design website, and then pulling that down to the robot. So it's really the same thing. If we go, okay, our protocol here is drawing. Our protocol here is something, maybe something as simple as passaging and making, archive stores of culture or something like that. The idea is that because of how we've written everything so far, this should be pretty easy to replicate. So we've got this one going at CCL. All right, there's another community lab. They fork it, and then they make another one, and then someone else, maybe they have a home lab, and they say, well, okay, I can fork this idea, and this will give me a lot of the tools I need to design stuff. And even a university, right? This is the sort of thing where a lot of different universities could spin up the same idea, and they have all the software, and they could do it themselves. And that's great. But it's also easy to collaborate. You can imagine that there's no reason that every single user needs to have their own version of the website. And so in this kind of model here on this slide, we imagine that there's a single website. So it's distributedcloudlab.org or whatever. And it goes a place where, I can submit a job, and then someone else can say, I've got some time for that. I'll run that for you. Yeah, I think sorry, I just glanced down two things. One, lab on a cloud, very good. I want to emphasize that this distributed cloud lab concept is very early. It's just kind of a tinkery idea, and yeah, I think we should find a really catchy name for it. And then yeah, Ray, you posted Transcriptic. So this is a concept, this kind of cloud lab concept has been floating around for maybe 10 plus years at this point, with varying results. Some places have had pretty good success with it. Other places have really struggled. Ultimately, from what I've been reading, the issue is that it's hard to do technology transfer. It's most science experiments are not plug and play. Excuse me. The reason, but that doesn't mean it can't be done. It's just you have to be careful. The reason that I'm particularly excited about this idea is that it empowers that kind of cloud lab transcriptic approach for DIY bio. So it's the sort of thing where, well, okay, I've just got to scrape together a grant for $5,000 so I can buy an Opentrons, and then great, now I'm one of the nodes in this. And you can imagine that lots of us get together and do this sort of thing. And now suddenly, effectively, there is a distributed cloud lab that exists all around the world, and that power can be tapped for good in one way or another. So that's the idea. Like I said, Distributed Cloud Lab is closing, and I think this is a good gift to go out on, because the idea is here to let's automate to help all of us. These tools, automation has a nasty habit of being a centralizer of power, and I don't think it needs to be. It's the sort of thing where we can actually all help each other, and so part of the bent of this project is to empower that and make it possible through all this open source and etc. Yeah, last slide. If you've been thinking about how to get involved or anything like that, through CCL, there's the mailing list, and I should have put the spreadsheet for signing up for that mailing list, and I did not. So I will find that and get that into the links. And then,
if anybody would like to be added to either the CCL Robotics mailing list or the CCL Art and Science mailing list, feel free to put your email in the chat and I'll add you. And let me know which one of those two.
Ooh, change slides accidentally. Yeah, so great. So there's the mailing list, and then, as I said, there's the GitHub, which is linked here, github.com/cclrobotics/artbot. That repo was named before I realized there are lots of things called ArtBot, but not that many things called BioArtBot, because there's a lot of things that just use like crayons and stuff. And of course, you can email me. I'm just at timdobbs@gmail. And yeah, I think I have a lot of work to do, honestly, about how to like make this a easier project to jump in on. It's been a long pandemic, man. I don't know what else to tell you. But yeah, I'm if you can get on this mailing list or whatever, I'll make sure that as I kind of get better about weekly meetings or having kind of easier places to jump in on collaboration, that you're made aware of that. Yeah, that was really all I had. There was lots of good talking here. I'm kind of curious about people's experiences with Transcriptic. I've never used them personally, but if there's any other kind of direct questions at this point, shout them at me.
Jessica, how much have you done with Transcriptic?
I did a single experiment maybe 4 years ago with them. I was actually just sitting here thinking to myself, what did I do with them? And I actually don't remember. I've wiped my brain clean when I left that job. Yeah, but I recall it went well, and we got results from it. So yeah.
Yeah, I've done a lot of reading with them. I've had jobs where the primary job is technology transfer. How do you take a process you did in one place and move it to another place? And there are so many ways for it to go wrong. And from what I've read, they've done a pretty good job at hammering out some issues. There was at least one paper that I'll have to dig up where Transcriptic sort of donated time to students to do experiments. And there were a lot of lessons learned is kind of the takeaway. It was not a failure, but it was a lot of lessons learned. So yeah, I'm a keen watcher of that because I think it all fits together. I think in a lot of ways, this is sort of modeled on being the nonprofit DIY version of something like that.
Yeah, I think we were a little bit, my experiment might have gone a little better is because we weren't moving a workflow because we didn't have the robotics capabilities. So we were just sort of, we relied on them a lot to come up with sort of the protocols and everything. And we were just using their robots to execute an idea that we had. So there wasn't technology transfer per se.
Great. Rami, I'm seeing in the chat, I think that's a great idea, the brainstorming. And then, but first I want to read this question from David here. To get purple, do you mix cultures that make red and blue, or do you need to get a plasmid that expresses purple? I don't know the exact answer to that, which sounds like a really fun experiment, actually, and is exactly the kind of experiment that I'm actually really hoping to encourage people who want to, learn from the platform, because it should be pretty easy to do to figure that out, right?
So we actually found that with the E. coli strains we were working with, we were able to blend them fairly well because they were all the same E. coli strain background. The only thing differing was which chromogenic protein they had. So they all grow nicely at the same rate and stuff like that. And the colors were fairly reliable. So I'll dig up some examples of some of the agar art that we did where people literally blended between all the different colors and worked just fine. If you're working with natural strains that are pigmented, then you're often going to have like weird interactions, or one strain might grow 10 times faster than the other strain, and it's going to go haywire. You're going to get very unpredictable results. But if it's all in the same E. coli background, yeah, it's nicely behaved.
Yeah. One kind of interesting thing we've seen from that is that if the pixels are quite dense, I say pixels, but I mean E. coli colonies, if they're quite densely packed, sometimes the middle ones don't grow that well. And I don't really have the physiological answer for why that might be. I thought maybe some kind of competition, but if you've seen blending work well, then maybe that's not it. Either way, I mean, that's exactly the kind of question that I would love to both answer for myself and, kind of encourage other people to answer using the tooling. Before we go on, I'm going to kind of close this screen share just so, so that I have you guys in a bigger window. If anyone still wants to write something down or whatever, speak up now before I close it. Great. Cool. There you all are.
Let me do a quick screen share of some of our agar art. I made a little collage from the last season that we did. And you see that there are some nice blends of the colors. Right, including this is the fluorescent one, and then this sort of blends into the blue. And yeah,
so,
you see some
Patrick, how are
nice
the blends achieved?
This was all using paintbrushes to apply the different strains, and they just went over the same area that they had already covered.
So you use physical paintbrushes with
yeah.
Mm-hmm.
I don't think they were intentionally like mixing like you would do on a palette with ink. I think they were just going over the same area that already had some strain on it.
Yeah, I wonder if you would get how your results would differ if you mixed the liquid broth together, 50-50 versus if you just did that technique you described.
I think because these strains are so compatible with each other, you can probably, if you know the cell counts in each, you can probably just do a 50-50 mixture and have it come out exactly the way you expect it to.
And when I was watching Tim's presentation, I sort of the thought came up too that you were doing sort of like biopixel art. Is it possible to make the instead of making pixels, making the stuff bleed in a way so that is there a way to do that using the robot? So you make it more blendy than pixely, is what I'm getting at.
Yes, but it's hard to do reproducibly. So you saw, I think that kind of early test we did, we used way more liquid than it turned out we needed. We were putting, I think it was like 3 or 4 microliters in each position, which gets really drippy and gooey really fast. But it was kind of a cool picture. And so, yeah, if there was a way to do that kind of purposefully, I think you could get there. I don't know. I've thought a lot about it. For a while, I was cutting paintbrushes and then jamming them into pipettes so that you could actually literally use a paintbrush on it.
That's a very cool idea.
It was medium successful. It's really hard to sterilize paintbrushes and
a Q-tip. Use Q-tips would be the way it swabs.
There you go. That's a good idea.
I like that idea.
So for agar art, we actually bought like this pack of something like 100 or 200 really dirt cheap, very fine paintbrushes on Amazon. It was something like 10 cents each per paintbrush, and it turned out they're autoclavable. So we actually wind up autoclaving each of them like 4 or 5 times before they started degrading.
Oh, that's not bad.
Yeah, that's pretty cool.
But yeah, I really think that's kind of my as far as expression, where I think like, well, what kind of drawing do I want to make? That is kind of the next place I go. It's like, well, I think a stroke is so much more interesting than a point. I guess
Well,
some
that's what
pointillist might
that's what I was
disagree.
kind of getting at when I was watching you in your presentation, right? Go more strokey than pixely.
Yeah,
So
well, it's
here's
a
a nice
hardware hacking problem.
example of some of the weird interactions you can get with naturally pigmented strains. So you see that one strain is actually just growing along the outside of the other strain here.
Yeah, I like that. That looks cool.
Yeah.
How
That one
did
is so
how
good.
did that happen again?
It's just Serratia marcescens does weird things. It likes to crawl along edges. Sometimes you'll see it like going all along the edge of the petri dish. This sort of spreading out here is it's actually swarming. You can kind of see where it was painted, but then it sort of goes out of control and starts swarming all over the place.
Yeah, right. Sort of like
What's the fuzzy
kind
stuff
of
on the
The
top?
fuzzy stuff is
Jackson
actually a
Pollock.
mold, because it took a while for whoever did this to make their design. And while you're doing it, you're having the plate open while you're hovering over it with a paintbrush. So you often get some contamination on there. There's some other contamination here. Yeah.
But you ended up getting something like Jackson Pollock.
Yeah. Yeah. There's like a spot that's the wrong color. That's probably contaminant as well, right there.
Yeah,
Another
because these are
way
natural strains, right?
Yeah, these are natural strains. Yeah.
Another way you could get them to be less pixely is just to grow them longer so they meld together.
Mm-hmm.
Yeah, I think we need to change the sort of printing protocol, at least somewhat, the inoculation part, because I've sort of found that they sort of grow themselves out after 2 days. I think maybe we start with a denser culture or something like that. But or maybe it's actually they need to be earlier. There's so many sort of cell physiology experiments that I think would be fun to do. But yeah, I mean the effect we've seen so far is it's about as good as they'll go, and then eventually the protein just starts to break down and they just lose color. But yeah, it's a pretty ad hoc procedure, honestly. The lab part is kind of just like, well, what works, and then I think there's a ton of space to like figure out how to do that kind of in a more make a more compelling result.
Is there also something you can do chemically to keep the protein from completely breaking down and sort of suspending it at a certain point?
Yeah, there's this group, Jeff Buca's lab at NYU. He might have moved universities, but I can't remember. But it's yeastart.org, and these pictures are mind-blowing. I just put it in the chat, but they use a LabSite Echo, which is like a $500,000 piece of equipment, and they get really, really high precision. And they have a piece of software that they very gracefully sent to me, and we've just never implemented. So if anyone wants to figure out how to implement it, that takes a photo and translates it into like a color map for the LabSite Echo. So they take these photographs and they turn them into these really beautiful yeast, growth things.
Wow, that's funny. I was just about to like say something like that. It seems like it's tangible to take a photo, put it to bitmap, and then reverse downscale the color to like 6 colors of the closest colors, and then translate that into a layer on the website that it could interpret. And there you have like an applet that can convert any photo to what works with the OpenTron, and it's all done by software.
Yeah, we talked about that. That there's some little routines out there that can subsample an image in terms of both the color space, but also the resolution. So you can get something off the shelf that would do that.
Yeah, I mean, I think it's a great idea.
There's a picture from yeastart.org right there.
If anyone hasn't gone, check out Michael's screen. It's really... Anyway, so I bring it up, one, because it's really neat, but two, because if you read kind of their about section, they do not chemical treatment, but they have this process where they let it grow, then they sit it at 4 degrees C for 2 weeks, and the colors brighten. And then I might be misremembering, but I think they do some kind of preservation on the agar at that point. But yeah, we haven't played with that really either. Yeah. But I kind of always wanted to try it because it seemed like a cool protocol.
And if you're working with chromoproteins, I mean, they're protein-based pigments essentially. Eventually, the proteins are going to degrade. But there's ways of working with natural pigments that actually will stick around even after the cells are long dead, right? So you can make it permanent. You just have to do a little bit more engineering.
Yeah. And I mean, it would also be cool to figure out a way to kind of make it more tangible to people who are only using the website and not in the lab. Like I talked about earlier, this idea of like, oh, well, can we set up more cameras basically and do like a telepresence? But like, it would also be neat to be able to send them art, like, but we can't send agar in the mail. I mean, that's not going to work. So we have to figure something out.
Or some automated system that tells them your particular design will get live streamed tomorrow at 5 o'clock. And
yeah. I've been playing around with oh yeah, the girl with earring.
The girl with the pearl earring. Yeah, exactly.
Yeah. No, lately I've been really excited about like live streaming technology and learning how to use OBS and everything. And boy, it's neat. They got a lot of good hooks there for like. I've been thinking about the CCL quiz show, and I'm like, oh no, I think it could look very visually cool. So yeah, I think there's a lot of ways to automate that as well and set up those kinds of systems you're talking about, Patrick.
Oh, speaking of Van Gogh, here you go. There he is himself.
Tan, I see you talk about like a rundown of the code. Here's what I'll do. Like I said, I kind of need to get off my butt on this. I'll send out an email to the list for sometime next week. I'll schedule a time to someone who really wants to look at the code. I was really struggling to like, how do you talk about software in a compelling, like a visually compelling way? And it's not look at code. So I didn't include it in this. But yeah, I think, like I said, I've been really enjoying it, and I feel like I'm learning a lot as we build it out. So I can send out an email for a time for meeting up and getting into the weeds on the software.
So the YeastStart website, they actually are using red fluorescent protein and a couple of other fluorescent ones, but then they also are using biosynthetic pathways for beta carotene, violation. So they are using a couple of pigments that should be much more hardy than the protein-based colors.
Yeah,
Is it just easier to grow?
Just as easy, I think.
I think violacin is actually secreted, so you could get some neat stuff happening with that.
Yeah.
I was looking at their Petri plates, and it looks like they have really consistent round dots that don't seem to bleed out.
Yeah, I think. So I had a call with the woman who was kind of running that. I don't know. That was probably more than a year ago now, but she was saying, yeah, a lot of that it's because it's this lab site echo, which works by vibrating nano droplets down into the plate, and so they're really, really precise in a way that the Opentrons has no hope of being.
Yeah, and then they probably time the plate exactly when to stop growth and put everything in the freezer, so the pixels don't get any larger than they intend them to be. But that's, all of that is a good amount of work to control that exactly. And this is what happens with our agar art as well. You have a window of maybe a few days where the picture is at its optimal. And after that, all the lines are getting fatter and the colors are starting to fade. And yeah.
I'm just wondering too, on the opposite end from nanoparticle vibrations, if you go all the way to like DIY, how might something like this translate into like a microbit, simple, maybe 2-color, you've got a servo motor and you're just coding in block code and microbit, and you're dropping, maybe only it's kombucha that's been colored with cactus fruit or carrot. So that you can have an expression, like a desktop at your home where you're kind of doing a simulation of the BioArtBot. You understand the mechanic side of it maybe a little bit more, and the connection between code and bio and all that. I think that would be kind of cool too.
Yeah, or even using like a little controlled CNC to carve a stamp out of a raw potato. and then use it
Potatoes
to.
were the original auger.
Yeah,
Is that true? I didn't know that.
yeah,
That's
yeah.
cool.
Slices of raw potato. It's good for certain organisms.
I do really like Corinne's idea because it gets to a very, very like similar or analogous kind of like coloring with liquids. And the part that's not happening, in front of you is the E. coli incubation. And so, that's the part that you can teach, but like the mechanics of it. Yeah, I really like that idea, Corinne.
Yeah, I agree. And I guess what do I want to say? That I come back to that simple and flexible idea is that we tried to make it specifically so that you could swap out what the robot was if you wanted to. So you just kind of, have to sit down and figure out how to map the, the sort of grid output that the website gives you into instructions for a robot. So someone has to kind of write a template, but once the template's written, it should be redoable. And yeah, I agree. I also really like the idea of making it even lower barrier to entry.
Yeah, because I'm just saying, like, I'm thinking about how you framed it as software, hardware, wetware. How can you replace each of those? Hardware, microbit, that's like $16 plus, some, sensors and server motors that are really cheap. Hardware, you could code this and, the block coding of microbit, or you could do it in Python and microbit. In terms of the wetware, you could move to drinkable kombucha, because I'm from the lens of like, I'm working with low resource teams that are in ag community, or they're in East San Jose, and like, how do I bring them into this conversation really deeply? And I think that might be kind of fun to just kind of push like what's possible on the lowest end possible, but then let them see what's on the highest end as well, and then what's in between. And I think it could be really a cool ecosystem with this conversation.
You could also do, have them go out and actually isolate naturally colored microbes from their environments, which is kind of another fun way to get them in low resource, because they're out there, they just grow and they're colored. So
yeah, that's cool. I think another challenge I have right now with my teens is like they're all in like multifamily households. They've got younger siblings, so creating a BSL1 spot in their houses, it's on their bed on a cookie tray. And then moving it to a shelf. And so, just staying with really like food, like something you buy off the shelf. But I love the idea of talking about microbes too. And I didn't know that about potatoes, Jessica. I thought it was so cool. So,
yeah, that's how the first salad was done. Somebody had a potato in the lab and stuff grew on it in colonies. And they're like, look at that.
What's a microbit you mentioned? Is that like a Raspberry Pi?
Oh my God, microbit's so awesome. It's developed by the BBC, and I forgot what other company in England. And gave one to, what was that?
I put the link in the chat.
Yeah, thanks. Yeah, it was given to every
microbial.org.
single student in the UK. Every student got one. And I think that was like in 2014 or something. Really for the reason that I was just describing, like how do you engage all students in this conversation with a very low resource, fun interface, friendly. You work through the whole interface. Like the moment you plug it in and you go to the website, it's playful. But it really it's the lowest barrier, I think. I mean, it's the tool that I think is most accessible to start getting youth into these conversations. Achieved. Yeah.
Yeah, this is super neat. I didn't know about this either. That's a great idea.
It's like an Arduino kind of thing, right?
Yeah, but
except more user friendly.
Yeah.
Yeah.
I mean, Tim, I feel like the conversation is like naturally like tilting towards like sort of like, age of COVID. So we have like distance learning, we have Zoom stuff, what can actually be, I like the idea of like, if in terms of working with E. coli, you have like a finite window where it's optimized. So it in and itself becomes an event when it like turns into something that you can see. So I think that's super cool too. We don't have to spend too much time, but I'm already hearing sort of like these threads of how to translate this to COVID times. It still might require setup and just kind of like thinking differently, since everyone can't be there handling the robot but I think there's lots of opportunity to think. And I'd love to hear what other people would think, like, Jillian, I'm gonna pick on you. I'm sorry. It's but you know, you're joining us for this, and like, if you wanted to use this, how could you think about ways, like, how could you engage with this from your own home? Like, what would be easy for you? Sorry for picking on you, and also you don't have to answer my question and say,
I don't really have any ideas, so.
That's totally fine. But yeah, like I would love to explore that.
Yeah, I agree. I think this is a great time and kind of space to brainstorm about ways to kind of engage with the project, or do something fun even though it's COVID. I mean, I'll start, and then I'm going to pick on someone, so get ready. I mean, the simple idea was something that we were talking about earlier. I was asking Patrick about like a live stream kind of situation where, someone goes into the lab and then just everyone else is kind of seeing their camera, which I think sounds pretty cool. So it's kind of like a virtual meeting, and like we can all, one person is the hands and everyone else is the brains. So I think that could be a cool way to kind of start doing something. Yeah, I guess I'm gonna ask Tan, just because I feel like I see your gears turning around like cool things to do. What are you thinking about? Any thoughts?
Well, I'd like to like see the machine in action. I think that'd be cool. Yeah.
Yeah.
So, yeah. I typically like learn best when, I sort of like have the hardware with me. I know that might be difficult with the age of COVID and all. But yeah. But like the slicer and all that it uses, it's like pretty standard CNC code, right?
It's we mostly use the OpenTrons API that sort of lays on top of the CNC code. We were writing raw CNC code for a while, and the API just makes life easier for that particular robot. It's so much less raw code that you have to write.
Okay. Yeah. Yeah.
So right now we have, as an example, we have open insulin going on in 2 labs, one in Baltimore and one in here. Probably David can speak to this much better than me, but you were saying, you have one hands-on and then you have the other brains. I'm not sure it has to be that confined. I actually think if you have a distributed network, I think like you were pointing out earlier, Tim, that you can actually switch back and forth between the hands and the brains. And I'd like to see that happening, for example, with the Open Insulin project between the Baltimore lab and the lab here. Hey, David, are you on? Can you speak to that a little bit?
Oh yeah. So Jan has in the past has I've had my cell phone with a video going and showing what I'm trying to do, and he's helped me that way. So he's available to remotely, in real time to show me what to do.
Yeah. So then it can be conversational and hands-on both ends. And, yeah.
You think it'd be easy to add a bitmap converter to the website?
Yeah. I mean, depends. For some values of easy, it's you probably can't do it in 15 minutes, but yeah, I don't think it's gonna be impossible either. And I think it's a great idea.
Yeah. Something I might be interested in trying my hand at. Yeah. So it gives you like a project to flex on to see if it, something that would work. And then with that implemented, you could make some pretty complex art, just like the yeast lab did. Yeah.
The only drawback might be that we get so many submissions that we can't process them all.
Yeah. I would just limit it to one per day. Yeah, just limit it to like one per day if too many people get excited about submitting.
I've given this a lot of thought because when it all started to click in my head, I remember specifically I was taking a shower, and as so often happens when you take a shower, like, oh, that's when the ideas start going. And I suddenly kind of got like, oh, the base of the code for that. I was like, oh, okay, I know how I would do that. And then I thought, oh no, there's going to be 10,000 submissions because it's so cool. And so the website is self-limiting. It has built into it a little message that says, I'm sorry, we have too many submissions this month, and then it resets.
Or you can just build a repository for it when you have too many submissions and store those away, and then over time, have access to those.
Or we can cheat and make simulated petri dishes, where we like add random mold here and there, and some spots are larger than the others, and
libraries ran into this whole issue when they first started having 3D printers in their spaces, and they had to let people know, like, you're in the queue, but it might be a month or two before you get your print. That's what some did. I know that's what the Sunnyvale Library did. And then there's also, I forgot the name of this, it's like the main 3D print kind of hub that you can join if you've got a garage makerspace. It's, Tim, is what you were talking about earlier. It could be your garage makerspace or university. People opt in to be printers of STL files that are uploaded to this particular site. I forgot what it's called.
3D hubs, I think.
Yeah, there you go. Yeah. So I think it's cool because you already know it's going to be popular, and like, what are the models that have faced this challenge?
Yeah, it's a
when you have somebody controlling it, you can just have multiple objects per plate, so you could stack your queues and interlace them. I think that's how they do it professionally, so that each run is just really efficient and high throughput. So
yeah. We also have talked about automating the process a lot more, because right now somebody needs to essentially prep the plates and get them in the right location and all of that. If we had something like a plate robot that could just take a pre-poured petri dish off a plate and put it in the right position for the robot, we could run through dozens of plates in a completely automated fashion.
So my experience with the plate robots is they tend to knock things over a lot.
Yeah.
So you have to use a very specific type of petri dish, otherwise it's a disaster. So they might not be as easy as you want them to be.
Well, for the robot, we have to use very specific plates anyway. We have to use the rectangular sort of a 96 well form.
Yeah, but it became even more like it had to have, because the different plate manufacturers have different little ridges that hold the plates in stacks, and it had to be exactly the right ridge, or the robot would just sort of knock them over. And they can be a disaster. So
especially if you expect them to behave automatically and autonomously. Yeah.
I honestly, I think there's a lot more really simple, like just procedural improvements to be made. Like in the video I showed, we are pipetting 5 mils of liquid culture into these 15 well, or these 6 well, I think they're 15 milliliter plates. We showed that you can do it with a 96 well plate and like a milliliter or less, because you need almost no liquid to inoculate.
Yeah.
And so there's a lot of things like that. I think that would also speed it up. My other thought too is that we got to play that game of how efficient do we get without over specializing to the point where it's not easily adaptable. We're not trying to be Ginkgo here. We're trying to be, well, my thought is, why not a million Ginkgos all around, or a million micro Ginkgos, I guess is what you'd call it. So I like the idea of like, okay, let's put in something like a plate handler, but let's make it modular. Let's make it so it's like an option.
So actually, Ginkgo might be interested in this type of thing. They do have a artist residence every year where they work with these sort of socio-biological art. Natsai, who spoke at Bio Summit this year, was their first fellow, and she did dyeing of cloth using printing with robots, and it was really cool for microbes.
Cool. As in like printer printing?
So it was basically the same type of robot, but they printed the colonies onto cloth, and it was like a low solvent, low liquid way to dye silks. It's really beautiful. I'll see if I can dig up some of her stuff quickly.
Yeah, that's very cool. I mean, as far as like long-term kind of like governance or anything like that around this project, I don't really know the answer. I can tell you short-term, like kind of where it settled on for me is that it did get some funding through a short-term fellowship. So I'm gonna go to Paris for an institute there for 6 months, which sounds really exciting and cool. And I'm still sort of trying to think about like, okay, what does that mean long term though? It's it's funding for a little bit to do a little more development, and kind of the sort of proposed outcome that I wrote in the application was that we get basically the product as it is, but it's a little easier to adapt. It kind of has some educational modules with it, and stuff like that. So I'm hoping that becomes a place for it to grow. But then, where do we go from there? I don't really know. And I think there's a lot of space for it to be kind of, it's a community project. I don't think it's my project, right? And so to figure out like, oh, well, how can we, get some more funding for it, or figure out how to like, how do people want to work on it? That kind of stuff, which is really open.
Tim, when does that fellowship start for you?
It's supposed to be April 1st. Sure hope the COVID numbers don't keep getting worse, but yeah, it's supposed to be April through October. So I'll go over there then.
Yeah, April things might be getting better.
Fingers crossed. I mean,
in,
we already delayed
France,
it once,
though, right?
so.
It's in France, right? So, I mean, how well is France doing with their vaccine rollout?
Yeah, well, I mean, that's the question. I don't really know. I haven't looked at their vaccine rollout recently, but
probably better than ours.
There is also visa work about like, Americans aren't actually allowed in France right now. You have to get an exception as a researcher, so it's a different kind of it's a whole thing.
Well, COVID's really bad there right now as well.
Oh, okay.
In Middle Europe in general, COVID is just as flagrant there as it is here in the United States.
So in the meantime, that's a thing that is coming down the pike. But in the meantime, I think there's still, if there's ever a bright side to all this lockdown stuff, it's that, well, it's an opportunity for us to figure out how to grow this kind of in it's on its home turf, right? I mean, this whole thing grew out of CCL, and I feel like that community is really the strongest resource for it. So we can keep figuring that out. We're at 8:30 now. I'm really enjoying the discussion, but I also don't want to, make people feel like they need to, sit around here all night if they've got some other plans for their evening. So is there any other thoughts people want to throw out there, kind of get into the mix right now? And if not, I promise I will write some follow-up emails. I think this is something I've been particularly bad about this year. And we'll figure out how to kind of make it something that is a little easier to jump onto, and we can all get involved, right? And also thank you. Thanks for everyone who came and kind of listened to what was kind of a lengthy talk. So I appreciate that as well.
Thank you, Tim. This was a lot of fun. Very interesting.
Yeah.
That was great.
Thanks.
Good session.
All right. Night, everyone.
Night.
Thank you.